skeletal, cardiac, and smooth. Okay, I gave you 3 things to remember about each one. How do you describe skeletal muscle? It is
Striated.
Voluntary, because you can tell it. It is striated and more than one nucleus in each cell because they're so large. How do you describe cardiac? Striated,
Involuntary. And uninucleated.
and one nucleus. And smooth muscle is
Not striated.
non-striated.
Uninucleated and involuntary.
There you go. Which one heals the best?
Smooth muscle.
And which one doesn't heal at all?
Cardiac.
There you go. All right. So there's that part done. Done. Okay, let's go to that. Oh, we're flipped in this one. Okay. Where's the picture? How come nothing is going on to the overhead projector? What the hell happened? Sunny beaches. Oh, and somebody's got that off too.
Involuntary.
From here. So, okay, how do we go about doing this with a
Well,
picture?
the time is right.
Can you
No.
just change your screen?
Yeah, it's gonna be, you gotta overlay these screens because here's screen 1, screen 2, and that's screen 3, and you gotta be like this freaking computer nerd to be able to do this. I'm like, son of a bitch, who would do this?
I upload them on
Okay.
YouTube.
For the muscle cell. What do you call the plasma membrane? Sarcolemma. And then we had, do you remember the blue stuff? Sarcoplasmic reticulum. What did it do?
Stores calcium.
Stores calcium. And then we had the sarcoplasmic reticulum. How did we get the nerve impulse from the sarcoplasmic membrane into the deep part of the cell? Transverse tubules. And then you had thickened regions of the sarcoplasmic reticulum that butted up to the T tubules, and those were called terminal cisternae. Okay. Then the functional unit of a muscle cell is the sarcomere, and when contraction occurs, what literally happens? Why do they call it the sliding filament theory? What's that? And that's what I'm trying to get out of you. We had 4 proteins that we talked about. Which 2 proteins slide across each other? What's the thin and thick ones called? Thin one is actin and myosin. So it is actually the actin and myosin filaments that slide past each other that cause the shortening of the sarcomere. Then what do we have the troponin and tropomyosin for?
Active sides of that.
For when
They're
the
regulatory.
head attaches.
So which one is it that actually covers the active sites?
Tropomyosin.
Tropomyosin. What's troponin's role then?
Helps move it.
Calcium comes in contact with it to move the head.
Binds calcium, changes shape, and then it pulls the tropomyosin off those active sites. All right, so there's our labeling part. So this applies to both nerve impulse conduction or nerve impulses and muscle contraction. Okay, what would be your resting membrane potential of a muscle?
Negative 90.
Minus 90, and if this were a neuron, then it would be
Negative 70.
minus 70. What is it? What is the name of the structure that actually creates the resting membrane potential?
The lead channels.
Lead channels. And then to keep it from equalizing ions on either side of the membrane, we have what that maintains it?
Sodium potassium pump.
Sodium potassium pump. Where was I going? Dumb shit here. Okay, so muscle contraction. Let's do this next because that's resting membrane potential. We want to pick it up at the axon terminal. What I want to do is practice your steps of muscle contraction because there's not going to be multiple choice questions in there because that's one of the questions that you're going to get in your essay. So let's do this.
I know, yeah, I was not necessarily.
What was the saying?
The ion gradients and so on, the action potentials.
It would stop it because they can, what's the word for it?
They go to zero. So all your ions would leak across the channel and pretty soon you'd all be equal across, so you wouldn't have any resting membrane potential. If you don't have a resting membrane potential, you cannot depolarize and happen. What is very similar to that we studied?
The pufferfish? TX?
That pufferfish
Something?
and Novocaine, lidocaine.
Oh.
Yeah, they're all using
was
that
so
same
confused.
process of block the sodium potassium pump. So multiple times now we have had that situation. All right, there's that. Okay. So we just got done practicing that. We did all of this. We did all of that. Okay. So there are three ways to alter the amount of tension in one fiber. What was the first way to alter tension in a single muscle fiber?
Size?
Recruitment, right?
We'll get to that down here, number 3.
Is it like the twitch? Muscle twitch?
You're getting past this point. Frequency
Oh,
of
actually.
action potentials, how fast they arrive. And what was the term that we used to describe that?
Oh.
Wait,
Or
summation.
nope, you're into nervous tissue.
What's the first letter?
Unfused
Infused, that's right.
and fused. Remember?
Yeah.
Because if there's still time enough in between your action potentials, you have a little bit of time to relax in between. So basically, what is causing this increase in tension? What are you increasing inside the cell? Not carbon, calcium. You're releasing more calcium. More calcium you release, the more cross bridges you form. More cross bridges you form, the more tension you can create. So it all goes back to calcium. And then here, they're coming so fast, you don't have any time to relax whatsoever. And then number 2 was, okay, am I like this? Am I like this? Am I like this?
The amount of stretch?
Yeah, they call that length tension relationship. And the basic line was, where do we generate the greatest amount of tension? Slightly stretched. Not overly stretched and not overly contracted. Because if I'm overly stretched, think about the amount of overlap. You don't have much overlap, so you can't make very many cross bridges. And if I am completely contracted already, then your actin and myosin filaments start bumping, your actin filaments start bumping into each other. And then the third way that we can alter muscle tension is type of muscle fiber. What are our 2 choices, Haley?
Diameter?
of this?
Yeah.
What's the difference in diameter between type 1 and type 2? Who's what?
One is
Type 1
smaller.
is smaller. Type 2 is larger.
Type 1 is
Who has more capillaries?
type 1.
Yep. Increased capillaries, which means decreased capillaries. And how are we going to make our energy if we have good blood flow? Oxidative catabolism. And then this one would be through all these terms. Oh my gosh. Glycolytic catabolism. And so, what are we not using here?
Oxygen.
We're not using any oxygen here. Which one's fast?
Type 2.
Which means that this is slightly slower. Which one has more oxidative enzymes? Yeah, more enzymes and fewer enzymes. Which one's called red meat?
Type 2. No, type 1. Type 1.
Why is type 1 red meat?
It has more
Because
blood.
we got more blood. And then we call this, think about the breast. What do chickens use their breast for?
Showing
Flying. How far can a chicken fly?
Not very far.
Not very far, because that's, one is more sustained and the other is for power. Which one's power?
1.
Type 2.
Type 2 is power. Remember, I'm flying, I'm a chicken. These breasts generate lots of power, but for a short period of time before they fatigue. So this is going to be more sustained, but weaker. Okay, does that ring a bell on type 1 and type 2 muscle fibers? So those are all the different ways that we can alter tension in a single muscle fiber. Now, we started talking about ways that we could alter tension in an entire muscle, not just a fiber, but the whole muscle. And that comes down to what? What is a motor unit? One neuron and all the different muscle fibers that it innervates. So if one motor unit is not enough to generate enough tension to get the job done, we recruit more motor units to that job, and that's how we get that done. So that is tension in a whole muscle. And this doesn't surprise me that you guys just didn't pick up on that detail as we were going through it. It's like it's clear as bell in my head, but it's like you're just like listening to all this crap, and I'm like, oh my God, does it make sense now how we're discussing two completely different things? Tension in a whole muscle versus in a single muscle fiber. Okay, any questions on that aspect? All right, that whipped through some stuff pretty quick. Oh, yeah. Oh. Okay, another thing that we talked about was, we actually talked about it before this. Let's see, how is that set up? That was like 2 seconds. And
Oh, yeah.
what was the next one? How many seconds?
10 seconds.
Yeah, because it's 40,
Pictures on my
10
phone.
seconds, 40 seconds, and then hours. Okay, so what did this represent? Stored ATP, but we can't store very much ATP because
We use
Dies
it.
quickly.
it's a very unstable molecule and it will fall apart. Then, how do we generate power, ATP, for roughly 10 seconds? Creatine phosphate, and we simply give that phosphate group to ADP to create ATP. Then the next round of energy comes from what can we do for roughly 40 seconds? Glycolytic catabolism. So we're just going to break a glucose molecule in half that only liberates 2 ATP for each glucose molecule. We can do that for a short period of time while we do not have enough oxygen available. But in the long run, what will last us hours? Oxidative catabolism. Yep. All right, so there's that. Creatine phosphate, there it is. And we already did the sequence that we use energy. So sugars, fats, followed by amino acids. Okay, so there's your muscle twitch. That's when we start talking about the twitch, was heading into how we increase the amount of tension. Okay, there's type 1, type 2, did it, done. Okay, myoplasticity. What is the basic difference between endurance training and resistance training? What will endurance training primarily target? Your type 1 muscle fibers. And these are going to be biochemical changes. So what's going to happen to a type 1 muscle fiber when you practice endurance training? More blood flow coming in, more enzymes to break it down. Okay, be more specific. What would it do to the size of a muscle if you were doing endurance training and you were hitting the type 1 muscle fibers?
Increase it a little bit.
You'll increase a little bit of size, but what you would mostly see is more muscle tone. So it will tighten it up. There's going to be more muscle fibers firing off, but it's not going to make them like huge or anything. What is another organelle which you're going to have more of on the inside of the cell to make your energy through like oxidative catabolism? Mitochondria. We're going to increase the number of mitochondria also, along with that blood flow. What is going to be your primary result from resistance training?
Bigger.
Bigger. And what is the word for bigger? Hypertrophy.
Oh.
And then, since y'all getting into healthcare, if you have a bedridden patient who is not able to use their muscle, what's going to happen to it? Atrophy. So, hypertrophy, get bigger. Atrophy, get smaller. Use it or you lose it. Done. And that's the end of the muscle chapter.